name is Robert DuFort. I am one of the field medical specialists with IDEXX. So we're going to talk primarily about what happens after the blue dot, and that refers to one of the reactions on the SNAP 4Dx. And I'm not an expert in vector-borne disease, and there are probably some of you out there that know a lot more about it than I do. So please bear with me. We're going to have ample time at the end of the presentation for questions and answers. So I think what we'll do is we'll hold all the questions until that point. So I'm going to go ahead and share my screen and we'll get started with the presentation. Yeah, this is really an apropos title, what comes after the blue dot, because these blue dots don't necessarily mean active infection. And with a lot of these tick-borne diseases, as we will discuss, the clinical signs may be inapparent. So the animals may come up with a blue dot. The owner may never have noticed the presence of clinical signs. And so the question is very good. What do you do after you get a positive on the 4Dx SNAP device? So again, I'm Robert DuFort. I'm a veterinarian. I'm a diplomat of the American College of Veterinary Internal Medicine, and I belong to the Global Medical Organization. So, you know, we're going global and we're all part of the same world. And again, that's what makes it a lot more exciting to be here with you today. So our learning objectives are as follows. We want to improve canine care through better understanding of tick-borne diseases. I know a little bit about tick-borne diseases in North America and virtually nothing about tick-borne diseases in South America. So a lot of what I share with you may or may not be applicable to your own country or your own region of the world. We want to overview specific test classifications and also talk a little bit about statistical terminologies used in the evaluation. We're going to discuss seroprevalence, pathogenesis, clinical presentation associated with these infectious diseases, and then we'll review the different screening and diagnostic tests as well as results and interpreting for improved patient care. So here's our workhorse. It's the 4Dx. And why do we even care about testing for tick-borne disease? Well, this slide says it all. Dogs are sentinels for tick-borne disease in the area that we live. And some of these diseases are zoonotics. So if the animal is infected with an agent carried by a particular tick, we can decide whether that tick is also carrier of a human infectious agent like Ehrlichia ewingii. And it makes for very valuable information for the MDs in our area as well as us. So we really are one medicine, one health, and we're kind of right in the middle of it being veterinarians. So public health benefits, co-infections. A lot of these ticks that carry these diseases can carry multiple infectious agents. And so we will want to know about that. And then testing also will want to treat if applicable, and we'll talk about the reasons for testing. And then probably as important as anything else, if we know an animal's been infected with a particular tick-borne disease, then we can really get on tick control. And because these animals can be infected multiple times, one infection does not convey immunity. And with the exception of maybe Lyme disease, there really are no vaccinations that I'm aware of that we can use. So screening dogs for tick vector disease is important because a couple of these agents, canine granulocytic ehrlichiosis and Lyme disease, can also cause infections in humans and can be quite devastating, quite lifelong infections. So dogs are sentinels for the presence of tick vector diseases in our community. and veterinarians need and want to know about what's happening in their communities. This is a shot of what happens in North America, in the United States, and some of these ticks have got they're prevalent from coast to coast, like the brown dog tick, where others tend to be more regionalized, like the black-legged tick. So I tend to know or try to familiarize myself with what are the ticks in my particular region. And let's see if I can get that laser to work here. So my region of the country is California. And so we've got the brown dog tick, and we've also got the American dog tick. And we've got a very nice agency that helps us track the seroprevalence and locations of these diseases in the US, and it's called CAPC, and that's an abbreviation for Companion Animal Parasite Council. And you can Google that and take a look and see the information. It's got a lot of information that may not be regionalized for you in South America and the Caribbean, but there's a lot of information on treatment of the infectious agents, information about the ticks themselves. So if you've never logged on to that particular website, it's a pretty cool place to be. Okay, and so again, our ticks can transmit more than one infectious agent. So, the Lone Star tick can transmit Ehrlichia ewingii. That's a human pathogen. The deer tick or black-legged tick can transmit Anaplasma and Borrelia. So 2 diseases, and it's not uncommon in the regions where those ticks are found, like in my part of the country or up in the northeast part of the US, to have a co-infection with a particular or a single bite from the animal. Brown dog tick can transmit Ehrlichia platies, which is regionalized in a central part of the US, and also Ehrlichia canis, which is pretty well widespread throughout. And then the American dog tick, again, Ehrlichia canis for those. So, if you've got a particular tick in your region of your country, and taking a look at this chart, you can see that that tick can transmit more than one disease to our particular pets. And so we need to be aware of that. So other couple of ticks here, amblyomma is a tick that's found in the Gulf Coast. Again, maybe you're seeing that in Latin America, perhaps not, but this is a new tick for North America. This is the Asian longhorn tick. And something that's very unique about this is here's the female here, here's the male here. The female does not need the male to produce live ova. So she can just go out and populate the woods or wherever she's found without the presence of the male. So they're going to be a very successful tick in transmitting our tick vector diseases. So what's the big thing? All of these factors have influenced the spread of tick-borne disease in the US, and I'm sure you're seeing the same thing. So urban sprawl, we're getting into the woods, we're getting into the jungles, and we're infringing upon the area that was natural to these ticks. Climate change, whether it's warming of the climate or whatever, we find that these ticks are surviving or staying active for a much longer period of time than. So we've got a very mobile world. People get on an airplane with their pets and take it from one continent to another continent. In the US, our deer populations are expanding, and they tend to be one of the reservoir hosts. And then relocation of pets, this is, again, this is a very common phenomena, particularly after disasters in the U.S. So after Hurricane Katrina, we got a lot of greyhounds coming up to all parts of the U.S. that came out of Louisiana and the southern area where racing greyhounds are prevalent. And ticks, their domain is expanding. So they're seeking broader and broader range for their particular living habits. Okay, so what are our options for testing then? We'll talk a little bit in depth about the SNAP device. Depending upon what the organism is, this is an immunofluorescent antibody stain. And so if we were looking for something like Lyme disease in the kidney, we could look for it with that methodology. You can actually see some of these organisms on a peripheral blood smear. We're not going to talk about any of the culturable organisms. And then the last thing that we'll talk about a little bit is a very sensitive, very specific test modality called PCR testing. And I see that that is available now in our Brazil laboratory. So those samples will have a little bit better turnaround time than we've had previously. So what is serology? Serology is looking for the presence of antibodies or antigen on a particular format. So not so much looking at the blood, but we're looking at the serum. So we can do it with the SNAP test, but also in the laboratories, we've got a laboratory SNAP 40X device. And so some clinics send their testing in directly to the laboratory. Certainly, it's another way of confirming it. Immunofluorescence testing is available only through the reference laboratory. And it's not quite as specific. It may be a little bit more sensitive because IFA looks for multiple antibody types. So we can be looking for an IgG or an IgM, but you may or may not have that testing modality available to you. So let's talk about our workhorse here, and that's the SNAP 40X. Some of the hallmarks are bidirectional flow, and what that means is that the solution is taken two directions across the sample capture device. And so second step is to wash off non-specific binding. So what this does is it results in a nice, clean, very specific staining of just what we're interested in, and that's going to be the antibodies and the antigens. And then we've got a substrate that highlights it with a dot here, and that makes it very easy for us to read our sample. So again, bidirectional flow, sample is conjugated mix, passes across the capture agents on two passes. It forces the sample back across for the second one. It provides ample or additional time for the binding of the antibody. Well, I'm talking about that. Here's a little test question for you. On the SNAP 40X, 3 out of the 4 of those test spots detect the presence of antibody. Only one of them detects the presence of an antigen. Which of the test spots is looking for the presence of an antigen? And those of you that have already come up, I will confirm your answer. The heartworm is the only antigen test. All the other spots are detecting antibody, which traditionally means just exposure to a particular agent, with the exception of maybe Lyme disease. So amplification comes about when the binding of the sample conjugate reaches an enzyme that lights up the spot or puts a color to that particular spot so that we can see it. It maximizes the signal from even a low level of a positive sample. So we've got everything that we need for a perfect test. And here's the 40X, and I kind of say tongue in cheek that this is really a 6DX because we test for 2 different species of Anaplasma, 2 different species of Ehrlichia. we test for Lyme, and then for heartworm infection. So we've got about everything we can put on that format, and I don't think we're going to see the evolution of a 5DX or a 6DX. We're just kind of running out of room on that format. So a little talk about when we expect to see antibody present. And antibody detection depends upon the tick infecting the host recognizing that antigen, either the antibody or either the antigen from the anaplasma or the Ehrlichia, and then giving it enough time to develop an antibody response. And so it's going to take some time. We're not going to get a positive the same day you see a tick on the animal. So here's the intervals for detection. And, since we're looking for a specific antibody from Lyme, it tends to be a little later coming up as a positive. But 2 to 3 weeks is kind of the average turning point. A reliable antibody, maybe we'll get a weak positive early on. If we get a little bit more time to develop that antibody, then we're going to be more reliably positive on our test. And so where do clinical signs come about? Well, a lot of times, like with Lyme, it's 6 weeks post-infection, whereas with anaplasma or Ehrlichia, we can see it very acutely. So the lesson here is that with these two, which is going to be the mainstay of our discussion, with anaplasma and Ehrlichia, we can have clinical signs weeks before we have a positive antibody test. Okay, so it'll come and go. And again, if we're just arbitrarily screening an animal, the owner is, say, unaware of any clinical signs and it comes up positive, then that's going to be confusing for our clients. And we're going to want to be able to explain why that comes about and how that comes about, and probably more importantly, what to do next. Do we treat or not? So PCR polymerase chain reaction is an incredible test. What it does is it takes one copy of the DNA from Lyme disease, Ehrlichia, anaplasma, even from heartworms, and multiplies it. Each time it multiplies it, it takes that double helix of DNA. By heating the sample, it splits it into 2 individual chains, and then the ingredients needed to make the complementary chain are in that reaction well. It's cooled again, and now we started off with 1 copy of DNA, and we've got 2. Well, each testing goes through 40 cycles. So if you take 2 to the 40th power, you end up with millions and billions, and actually trillions of copies of the particular agent that we're looking for. So it's highly specific for the exact DNA or RNA that we're seeking. Although it's highly sensitive, a negative result does not rule it out. Okay, there may not be any particular sample in the blood sample that came into the laboratory. So this next slide gives an idea of how the cycling works. So each one of these occurrences along this horizontal graph is a single sample, is a single cycle, I should say. And you see with any particular patient, every time they move to the right, it's another cycle, and the amount of DNA that is produced doubles and doubles and doubles. It gets to an extinguishing point here because there are no longer enough reagents for the polymerase to continue replicating. So this may be a little complicated, but this particular sample rises very quickly. It rises in a lot fewer cycles than this particular pet down here. And the reason is that he starts off with more DNA. So when he starts to double it, if he started off with, let's say, 100 times more DNA in his specimen than this one showed, it's going to take less time, less cycles, for him to exceed the ingredients in the test tube, if you will. So also, because of the ability to differentiate these, some of our PCR tests can be quantified. So we can actually, with some infectious agents, and this is more important these days with like an FELV infection, we can actually say how much the particular agent is present in the animal. And this is valuable for researchers and sometimes even for treatment. So let's compare serology and polymerase chain reaction timing then. So serology is looking for an antibody with the exception of heartworms, which is looking for an antigen. So it's useful as a screening test and also as a diagnosis for acute or active infection like Lyme disease. So here's a limitation. The animal will have clinical signs before that detectable antibody is produced. Okay, so there's a drawback. So you can't go in and jump in and do a 40X the minute you see a tick on your pet. You're going to have to give that animal a period of weeks to develop the antibody to give us a positive test result. Well, that's not so with PCR testing. PCR is not just looking for an antibody, but it's looking for DNA or RNA from the pathogen. So it's definitive for Ehrlichia or Anaplasma being in that particular pet's blood in that circulation. So it does indicate an active infection, and following it can indicate the resolution of the infection following therapy. Again, just because you don't get a positive PCR doesn't mean that the animal is not infected. It may be that there are inadequate numbers of RNA for us to really detect that infection. So tick-borne disease is kind of a pain. They initiate a hypersensitivity response. Generally, this is type 3 hypersensitivity where we've got antigen and antibody complexes. So the antigen binds to an antibody in the pet, causes a fairly large molecule to form, and then it gets filtered out in the filtering apparatuses of the body. So that's the kidneys, eyes, joints, and the central nervous system. Okay, so they are screening tests and they help us determine exposure to ticks and moreover exposure to the particular pathogens that we're looking for. So what's a veterinarian to do with a blue dot in a healthy pet? Well, what the blue dot does for us is it finds that the animal has been exposed. So if it's a backyard dog, then we know we've got ticks in the backyard. If the pet's been taken out into the woods for recreation, then we know that's where it's getting infected. So it opens the discussion for further diagnostics, opens the discussion for flea and tick control, and then opens the options for screening or rather treating our particular pets. Okay, a lot of these tests are accompanied by proteinuria. So we'll talk about the clinical signs for each of those agents when we get there. But because they develop these immune complexes and they're big molecules that get filtered out in the glomerulus, then that sets up the chain for inflammatory disease or an inflammatory reaction in the glomerulus, causes a leaky glomerulus, and causes protein to be dumped into the urine. So on some of those algorithms that you see that helps us with directions in pursuing the diagnosis and workup of a case, a lot of them do require a protein or urinalysis looking for proteinuria, and then sometimes even a urine protein creatinine ratio to verify the amount of protein that we're seeing. So here's that immune complex I was alluding to earlier. This is the antigen. So an antigen is like a protein from the anaplasma or a protein from the organism of the Ehrlichia. It's a foreign agent to our pets or people. And so we develop an antibody response as a way to clear it or limit its distribution. So they form these antibodies, and they're trapped non-specifically in the filter. So it can cause glomerulonephritis. Some of these can cause SLE and serum sickness. So this is where a lot of the disease comes from. And again, these immune complexes can be persistent, and that's why Lyme disease in a human can be a lifelong problem, because the complexes get filtered out, but there's no way of getting them cleared from the kidney, for instance. Okay, I'm going to go ahead and manually advance here. Whoop, I want to go back. Go back a couple.
Take your time. I think so far it's going very well. The pacing, the rhythm, it's very good.
Thank you very much. Okay, so I work for IDEXX because I believe in the work IDEXX produces, very, very sensitive and specific tests. And so, if you've got a sales agent coming into your practice and trying to sell you a specific test, don't just take their word. Don't just take their white papers. Take a look and see if they can provide you with a peer-reviewed, published reference that shows in print that these facts that they're trying to convince us of are true. So this is a study that was put into a veterinary journal that compares the sensitivity and specificity for the SNAP 40X with regards to the Borrelia and with regards to the Anaplasma. So the comparison here was done with a Western blot as the gold standard, and here with a IFA as the gold standard. So a SNAP was tested against an Acuplex, and here's the numbers. And we probably wouldn't be showing this slide if we were not superior. So we're pretty proud of our work, and don't believe me, I get paid by IDEXX, but take a look at some of these peer-reviewed references, and when our salespeople come around and hand out the literature, you'll have a reference here, and you can go online and read the facts for yourself. And I think that's very telling. Again, confidence in the results, and another peer-reviewed literature. Again, comparing the SNAP 40X with the Acuplex. So we're not here to bash our competition. We're here to show you in print the validated results of comparison studies that have been done. And again, here's just another comparison of the SNAP 40X with the VetScan test. So those studies will be made available to you by your IDEXX veterinary diagnostic consultant. So let me just skip over that, and we'll jump right into Anaplasma then. And Anaplasma, positives for canine Anaplasma. And again, dogs are sentinels for human disease. And you will probably see the same thing in the Southern Hemisphere. So that here's the reported incidences of canine Anaplasma, and here's the distribution for human Anaplasma. And you see the concentrations in this area correlates with the concentration in this area. Again, here's the concentration here, and this is the part of the US that is very highly affected with Lyme disease. So co-infection, yeah, here's going to be a co-infection with Lyme disease and with Anaplasma. Anaplasma can affect humans, as Lyme disease can also affect humans. And so our pets provide a lot of sentinel information for us in the management of not only pet disease, but also human disease. Not going to talk about Anaplasma platies, and in fact, it's a fairly regionalized disease in Middle America. I've never seen a case of it in my part of the country. And so Anaplasma is, Anaplasma phagocytophilum is a anaplasma organism that infects neutrophils. And when we used to see this in the laboratory, it looked a lot like Ehrlichia equi. And so for a long time, it was called Ehrlichia equi. And then the genome and the sequencing and the DNA of these particular agents were studied and refined, and we find that it is actually a specific. So acutely, what does it do? And these clinical signs here, particularly right up front, are going to be very similar, whether we're talking about anaplasma or whether we're talking about Ehrlichia. And these diseases can come in several phases. So it can be acute, it can be subacute, and it can be chronic. And if we can treat an animal with acute Ehrlichia, and I'll get to that slide in a minute, then maybe we can keep it from going to the chronic infectious phase, which is very often fatal with a pancytopenia. So fever, anorexia, lethargy, polyarthritis, and lameness. And in some cases, because we've got a meninges surrounding the brain, and it's a filter, these immune complexes can be deposited in the meninges, and so neurologic signs can be a part of the presentation for anaplasma. So in my part of the country, it's transmitted by the exotic species, Ixodes pacificus out here. With regards to clinical pathology findings, most all the tick-borne diseases, if they're going to cause a cytopenia, thrombocytopenia will be the primary abnormality that we're going to detect, followed by a lymphopenia. They may or may not result in increased liver enzymes. I don't see that all that commonly. So what's the take home here? Well, if they can cause some laboratory abnormalities, and particularly if they can cause some thrombocytopenia, what's one thing that we may want to do after we get a blue dot? Well, maybe we'll want to do a CBC. Or when I talk about Ehrlichia later on, you'll see that Ehrlichia also is coincidental with kidney disease. So with an Ehrlichia infection, maybe we want to do the CBC and a chemistry panel and a urinalysis. And so what this is doing is doing additional testing for us to determine the extent of the infection so that we can properly treat our pets. Okay, so we name those before: lethargy, anorexia, lameness, joint swelling. Thrombocytopenia, lymphopenia may be increased in liver enzymes. The animals may appear healthy, particularly if we're picking up in the subclinical state. And here's where the conundrum comes about. The subclinical state is very often where the animal is residing when we get that positive blue spot. So we get that blue dot, the animal's asymptomatic, and what do? And it's a great question. And we've actually had the Internal Medicine College try to come up with some consensus statements with regards to what's the best approach to an animal that's got a blue dot that is asymptomatic. And you know what? They could not come up with a consensus. So the jury was out on that particular endeavor, and that's too bad because, well, it's good and it's bad. It's bad in that it doesn't give us a lot of direction, but it allows us quite a bit of leeway to decide for ourselves exactly what we're going to do with that. So chronic cases, we don't really know what happens in the chronic cases, okay? Experimental infections have shown that prolonged chronic infections are possible, but there's no evidence to prove that chronic disease state from a persistent anaplasma infection is something we see in clinical practice. Okay, so here's a peripheral blood smear. This is may be a little difficult to tell. It looks like it's got a little segmentation right there. This is actually a monocyte. So we're finding these morula. These are the anaplasma organisms in the cytoplasm of the neutrophil. And again, previously we would call that Ehrlichia equi. Whole different genus, whole different species. So here's the Ehrlichia, and these are monocyte cells. I'm sorry, this is a neutrophil. These are the monocytic cells here, and these monocytic cells then are containing the Ehrlichia moruli. Here's a neutrophil here. So yeah, my first blunder was calling this a neutrophil. I should have stuck to my guns. So neutrophil with inclusions makes it anaplasma. Monocyte with inclusions makes it Ehrlichia. So sometimes you can find those moruli on a peripheral blood smear, but the numbers are usually pretty small, and it's pretty uncommon. And it used to be something that was fun when I was working in the laboratory, because the techs that look at hundreds of blood smears throughout the day would get so excited when they'd find these morula in these white blood cells and bring it. So they're also difficult to distinguish from other intracellular structures, and some stains can cause particulates and other cell pathology. So take a look at it, and if you see it and you're comfortable with it, call it, but you can also send those slides or that blood sample into the laboratory, and I would probably send the slide, because you may have just gotten lucky to find it on your particular smear. Send that smear in to us. So the timing of the moruli may also be quite transient. It tends to be in the acute stage. And then here's another Ehrlichia. This is an Ehrlichia that infects neutrophils. It's the Ehrlichia ewingii. Okay. We cannot differentiate Ehrlichia ewingii from anaplasma morphologically. We need to do PCR diagnostics to make that differentiation. So diagnosing anaplasma phagocytophila, IFA shows that the neutrophils are infected, good sensitivity, but reduced specificity. The SNAP test is excellent as far as distinguishing that peptide. So it's got good sensitivity and good specificity. And again, if we went back to that 40X slide, that 40X picks up not only anaplasma phagocytophilum, but also anaplasma plates. And so if you're in an area of your country where you've got plates, then you need to do PCR testing to differentiate those two infectious agents. Okay. Treatment. So the treatment is typically with doxycycline or fluoroquinolones, supportive therapy as needed. And usually the response to therapy is pretty quick, within a day or two. And recheck within a week or two usually indicates resolution of the thrombocytopenia and lymphopenia. So got a little case here. This is a lady, 2-year-old Shih Tzu, who came in with generalized pain, difficulty walking. Boy, the only thing we need here is a little fever, right? Pain on examination, particularly localized in the joints. So she had a hot spot on the back of her head. Maybe that was a tick. No fever here, but that's all right. We may have missed it. So the diagnostic plan was following diagnostics. Particularly with her clinical signs, we went forward with a 40X. We did the chemistry panel, electrolytes, CBCs, so minimum database was accomplished there. And what we found out is that she came back positive for anaplasma on her 40X. The owner elected to pursue treatment. She was started on doxy, and she responded. Then the important thing that we can recommend to our client is that here is the dog that was exposed to ticks. So now we have an opportunity to discuss tick control with our pets. These algorithms are available through your veterinary diagnostic consultant, and your algorithm is exactly like Mine is here. Moving on to Ehrlichia. Again, pretty wide distribution. We see more Ehrlichia in the US than we see heartworm infection. So the pathogen is Ehrlichia canis, infects the monocytes. Clinical signs looks kind of familiar, doesn't it? Fever, lethargy, anorexia, bleeding disorders. Now, in the chronic phase, they can cause a pancytopenia. So we'll get a thrombocytopenia, anemia, and a leukopenia too. Sometimes they can cause a hyperglobulinemia. Again, antigenic stimulation to the immune system causes globulin production, causes the presence of a hyperglobulinemia. So sometimes we start with a blue dot and work towards our minimum database. Sometimes we start with a minimum database and head towards the blue dot. So it's bidirectional here. So we've got an anemia. One of the things we do routinely in the US is do a 40X on it, particularly if there's an anemia and thrombocytopenia. If there's a hyperglobulinemia, we may do an electrophoresis, but we may also start looking for tick vector disease. And Ehrlichia canis then is intracellular, infects the monocytes, because it lacks these peptides, it evades the immune system, and maybe that's why Ehrlichia infections so commonly go to chronic infections, and those result in a pretty poor prognosis. Acute illness, subclinical, chronic phase of the illness. So this is pretty routine in all our tick vector diseases, but look at the similarities between clinical signs, between laboratory results here. I wanted to get to one slide before I open it up for questions that, I'll leave that to another day. I wanted to show you something that we found in a big study in the US. We found that the incidence between tick vector disease and SDMA testing positivity came out with a pretty astounding correlation. So we had animals that had chemistry panels and that also had a 40X present. We did their chemistry panels, and what we found in that comparison is that if an animal was positive on the SNAP 40X, it had a 112% increased incidence of kidney disease. Not quite as high with Lyme disease, and so this is studies that you can look at yourself if you go to idexx.com/kidney chronic kidney study, chronic kidney disease study. And again, just put into a graphical form. If we have an animal that's got a blue dot for Ehrlichia, that animal is also potentially predisposed to kidney disease. So a lot of correlation between the two. So what comes after the blue dot? Well, here's an algorithm for you, and it starts with a positive result. Then what do you do next? Well, it depends upon whether there's clinical signs or no clinical signs. Then no treatment recommended, treatment recommended. So take a look at this, keep it in your clinic right next to your 40X devices so that you can then decide what the best course of action is for your particular pet. And I think we're just about at the end of the slides. And so I don't know whether we can open it up for questions here, since this is an English-speaking audience. Here's the key points that I want you to keep in mind. Educate the clients about tick-borne disease and tick prevention, and how important it is to remember that these tick vectors are also transmitting human monocytic ehrlichiosis. So it is One Health. We can advise monthly ectoparasite control, and we can screen annually for effective prevention program. And I think I'll go ahead and call it quits there. Well, thank you all for being present. You've got some local talent in your particular part of the world that is more than willing to help you with taking it further, providing you with additional information. And it's been my pleasure to spend an hour with you and bring you what I know about tick vector-borne disease. Thank you very much and have a great day.
Os testes SNAP são desenvolvidos com a mesma tecnologia ELISA, usada em laboratórios de referência. Ao contrário dos testes de fluxo lateral, que são uma categoria inferior de teste, cuja tecnologia não se compara ao método ELISA de um laboratório de referência. 3 exemplos em que fazer um SNAP pode mudar tudo. E não fazê-lo também. Fluxo bidirecional. Apenas um teste rápido possui. O fluxo bidirecional melhora a sensibilidade, dando à amostra 2 oportunidades para se ligar aos anticorpos. O fluxo unidirecional pode prejudicar a sensibilidade, porque a amostra só tem uma chance para se ligar a anticorpos. Fase de lavagem integrada. Apenas um teste rápido possui. A etapa de lavagem integrada dos testes SNAP maximiza a especificidade, eliminando os resíduos na janela de leitura dos resultados. Não ter uma etapa de lavagem pode sacrificar a especificidade, porque os resíduos permanecem, permitindo resultados falsos positivos. Amplificação. Esta é mais uma razão para os testes SNAP ganharem sua confiança. A amplificação maximiza a sensibilidade, permitindo interpretar até os resultados positivos de baixa intensidade. Por isso, com o SNAP é mais fácil e confiável tomar a decisão certa para o seu paciente. Não ter uma etapa de amplificação pode sacrificar a sensibilidade e o resultado ficar difícil de interpretar, especialmente em amostras de sangue total. Um SNAP pode mudar tudo.